Hi friends, today we are going to discussing about one important concept in cellular regeneration, that is totipotency. So what is totipotency? And our plant cells and animal cells have this totipotency. So if you take a cell, that is plant cell, these plant cells can generate entire plant. If the plant cell or any cell generate entire plant or produce entire plant, this property is known as totipotency. A single cell can generate entire organism or plant is called totipotency. Similarly, if you take animal cell, it also can generate entire organism. It is also called totipotency. That means if any cell can generate entire organism, that is called totipotency. In case of animals or plants, if any cell can generate entire organism or entire plant or entire animal, this property is known as totipotency. So totipotency is the ability of living cell to express all its genes because every cell contain gene or DNA in its nucleus. So these genes express all required characters to generate whole organism from a single cell. This is called totipotency. So totipotency is the ability of all living cell to express all its genes to regenerate a whole new individual or organism. This is called totipotency. Generally, in case of plants, all the isolated cells extracted from leaf, stem, root, etc., any organ exhibit totipotency. In case of plant, whatever the cell you take, that cell can exhibit totipotency property. That means whatever the cell you extract and use it for culture and can generate entire plants. So that's why all plant cells are totipotency in nature. So whatever the cell you take, all the plant cells are totipotency in nature. Whereas in animals, only zygote exhibits totipotency. Whereas animals, not all cells exhibit totipotency property. Not all cells produce entire organism in animals. Only zygote exhibits totipotency property. What is zygote? It is the fertilized product of sperm and egg. When egg fused with the sperm or sperm fused with the egg, that is called zygote. The zygote only exhibits totipotency property in case of animals. Remaining other cells or all cells not exhibit totipotency property. Whereas in plants, all cells, whatever the cell you take, that cell exhibit totipotency property. That means whatever the cell you take, that can generate entire plant in case of plants. Whereas in animals, only zygote can exhibit totipotency property. Only zygote can only produce entire organism or entire animal or entire human, but not all other cells. This is the difference between plants and animals. All plant cells have totipotency property, whereas only zygotic cells have totipotency property in case of animals. Why all plant cells only produce entire plant, not why not all animal cells? Because to take this is a plant cell, these are the different types of plant cells. Whatever the cell you take can produce entire plant. So whatever the cell you take, either from xylem or phloem or sclerenchyma cells, chlorenchyma cells or parenchyma cells, epidermal cells or stomatal cells, so whatever the cell you take, all the plant cells can generate entire plant. So all the plant cells are totipotency in nature because all plant cells have plasticity, whereas very few or limited animal cells have plasticity. So why all plant cells can generate entire plant means because all, whatever the cell you take from the plant, that cell has plasticity nature. Whereas if you take animal cells, only zygotic cells have this plasticity nature. That's why all plant cells produce entire plant, whereas very few or zygotic cells only produce entire animal. What is plasticity? Plasticity defined as the capability to de-differentiate. De-differentiate means backward step. For example, already one cell derived. Then again, it can back to the original or mother cell type. That means already differentiated cell can come back to its original state and can generate entire plant. That is called de-differentiation. Differentiated cell coming back to initial state. That is called de-differentiation as zygote and re-differentiate as a whole organism. This is the plasticity. This plasticity exhibited by all types of plant cells. So what is plasticity? Plasticity defined as the capability of a cell to de-differentiate as zygote and re-differentiate as a whole organism. This type of plasticity observed in all types of plant cells, whereas plasticity observed in only zygote in case of animals. This is called plasticity. So individual cells of plants retain the ability to give rise to a complete plant because all plant cells have this plasticity nature. They can de-differentiate and also again regenerate as a whole plant. That is called plasticity. So the totipotency property of plant cells, all plant cell is mainly due to its plasticity. Because of its plasticity, whatever the cell you take from the plant, that cell can generate entire plant or entire plant. So this is called totipotency. Whereas if we take animals, these are the sperms and the egg. When one sperm fused with the egg, it can generate zygote. And this zygote has the totipotency property. The zygote only has totipotency property, and only the zygote can produce entire organism. Remaining all cells cannot produce organism. In case of animals, only zygotic cell has the plasticity and zygotic cell has the totipotency property. So in animals, only zygotic cell can produce entire organism. Why it is like that? Because once egg fertilized with the sperm, it produce zygote, and the zygote difference further division taking place, and it has totipotent stem cells. Once division, again further division taking place, it gives blastocyst containing pluripotent stem cell. The potential is decreasing. Totipotency means entire organism produced. Pluripotency means only few cell types produced. That means here pluripotent, that means the potency is decreasing step by step. Then blastocyst containing pluripotent stem cell further divided into isolated pluripotent stem cells, hematopoietic stem cell, neural stem cell, mesenchymal stem cells. Again, these differentiated stem cells produce limited type of cells. That means from embryo to uh, adult organism, their potency is decreasing from totipotent to pluripotent to pluripotent to multipotent, multipotent to unipotent. Because of this diminishing or decreasing of their potency, the other types of cells cannot produce entire organism. In case of, for example, if you take cells from hair, hair cells can produce only hair follicles, not other types of cells or entire organism. If we take cells from kidney in human, only it can generate kidney, but not all entire organism. Except zygote, remaining all cells of human or animal cells cannot produce entire organism because their potency is decreasing during development. So that's why there are different types of cells present: totipotent stem cells, pluripotent stem cells, multipotent stem cells, unipotent stem cell, like that. So only totipotent stem cell, that is zygote, can produce entire organism. This is the limitation in animals in compared to plants. Why it is like that? Generally, in plants, all cells, whatever the cell you take from the plant, it can generate entire organism. At any stage you can take, it can generate entire organism. If you take a cell from the leaf, if you take cell from the stem, if you take some from the xylem, or if you take some cell from the phloem, or if you take cell from any root also, it can generate entire organism at any stage. But in animals, only embryonic stage, that is zygotic stage, only produce entire organism. Remaining all cells cannot produce entire organism. Why it is like that? Both are living organisms. One is plant and another one is animal. Why plant cells can generate entire organism? Whatever the cell you take, it can generate entire organism. Why not in animals? Because pluripotent nature of plants is due to simple developmental stages. If you take plant starting from zygote to plantlet, it has very few stages of development. 3 or 4 stages are there. But in case of animals, very more number of stages are present. That is complex development is present in case of animal. Because of this complexity or large number of developmental stages, the totipotency property of animal cells is limited to only zygote. But in case of plant cell, if you take its developmental stages are very simple and very few stages are there. That's why plant cells exhibit totipotency property, whereas animal cells, only zygotic cells can produce exhibit totipotency property. Another. Reason is high degree of differentiation in animals, whereas in plants, less in differentiation in plants. If you take plant cell, you can find hardly some 20 to 30 different types of cells only. Whereas if you take animal cells, nearly 200 different types of cells, 200 different types of functions present in animal cells. If you take kidney cell, liver cell, RBC, WBC, like that, there are different types of cells present, nearly 200 different types of cells present. Whereas in plants, nearly 20 to 30 different types of cells are present. So animals have high differentiation, whereas plants have less differentiation. So less differentiation, because of this less differentiation nature of plants, they can easily regenerate or easily de-differentiate and regenerate into whole plants. Whereas in case of animal, they are highly complex. There are different types of cell, highly differentiated cells. So that's why it is not possible to the animal cells to easily de-differentiate and produce entire organism. So because this is another reason for why plant cells producing and whatever the plant cell you take, it can produce entire plant because of this less differentiation nature. And third one is complexity of animals than plants. If you take the animals, they are highly complex compared to plants. If you take animal, there are several hormones are there, but in take a plant, there are only five phytohormones are present. But animals, there are so many hormones are present. These hormones have effect on the individual cells. But in plants, only five hormones are there. So because of this less complexity also, plants can regenerate. Then epigenetic changes. What are epigenetic changes? There are different changes. Mutations are one type of changes in DNA. But epigenetic changes are not mutations. They can only chemically modify the nitrogen bases. For example, if we add methyl to the adenine, that is called epigenetic changes. If you add acetyl to the adenine, this is called epigenetic changes. Like that, some external groups are functional groups added to the already existing nitrogen bases and modify the gene expression. This is called epigenetic changes. These epigenetic changes in animals are irreversible. Irreversible means once one epigenetic change happen in the animal cell DNA, then it is not reversible and it gives one particular character to the cell. So, because of this irreversible nature of epigenetic changes in DNA of animal cells, this is another reason for lacking of totipotency nature in all cells. Whereas in plant cells, these epigenetic changes are very simple and these are reversible. So, these are the different reasons for why all plant cells have pluripotent nature, why not animal cells present process. Because of this pluripotent nature of the plant cell, they are using plant cells are extensively using in the plant tissue culture and plant biotechnology applications for the production of different varieties of plants. If you take this plant, this is plant. If you isolated cells from this plant, or if you isolate leaf and from the leaf cells isolated, then these leaves are cells cultured in the culture media, they can produce plantlets. So during short period of time, we can generate large number of plants. This from this in plants. This is called mainly because of totipotency nature of the plant. Second one is meristematic culture. Meristematic culture is mainly used for the virus-free plant production. Meristem means the end of the plant shoot. So older leaves are removed from the plant and the new plant, the growing apex region of the plant is taken from the culture. So these cells are called meristematic cells. These meristematic cells are highly differentiation potential, highly dividing potential. Because of that reason, so virus not infected to these cells. So because of that reason, meristematic culture is mainly used for the virus-free plants production. Another one is protoplast culture. Protoplast means if you take plant cells, it has cell wall. If you remove that cell wall from the plant cell, that is called protoplast. So plant cell without cell wall is known as protoplast. This protoplast culture mainly used for the genetic manipulation. So how we can perform this protoplast culture? So young plant, we take young plant from this, isolate one leaf as X plant, sterilize it, and peeling of the epidermis. This peel contain different types of cells and separate these cells and treatment with enzymes. After treatment with enzymes, the cell wall is dissolved and protoplast generated. What is protoplast? The cell, plant cell without cell wall. intact plasma membrane is present, but cell wall is absent. Then this protoplast used for the genetic manipulation. After genetic manipulation, again you need to generate cell wall. So cell wall generation, then callus formation taking place. This callus again differentiation into plant. So whatever the plant you want to generate, you can generate. So generally in plant cells, genetic manipulation is not that much possible because it contains hard cell wall. If you remove that cell wall, genetic manipulation is somewhat easy. Because of that reason, protoplast culture is taking place. This protoplast culture is also mainly because of the totipotency nature of the plant cell. So because of this reason, as all plant cells have this totipotency property, we are extensively used this property in plant tissue culture and generation of different types of plant varieties in animals. So this is all about totipotency.